Importantly, to start, I'd like to acknowledge that we're hosting this session on the unceded and ancestral territory of the Coast Salish peoples. This includes the territories of the Musqueam, the Squamish, and the Tsleil-Waututh nations, and the Métis Chartered Community of the Lower Mainland region. Importantly, you from all around the province are in your different territories, acknowledging the land, the people on it, and endeavoring to learn more and take better care of both the people and the land. So thank you all for that. Province-wide rounds is a collaboration between the University of British Columbia and BC Renal, supported with generous unrestricted educational grants from our industry partners, but also supported by all of you at the different health authorities, both by listening, but also many of you present. So thank you very much for making province-wide rounds as robust and interesting as it is. So with that, I'd like to introduce Dr. Pouneh Dokouhaki, who is a visiting locum pathologist from Saskatchewan with an interest in renal and kidney pathology and extra training in immunology and pathology. And she and I were just talking a few minutes ago that complement is the new thing in the kidney world. And so we will have a number of different conversations about complement over the course of the next little while. But she's going to speak to us today. And thank you for making the time and sharing your wisdom with us about complement system and the kidney, whether it's mission protection or destruction. So over to you, Pouneh, and look forward to having your insights as well as conversation afterwards. Thanks.
Well, thank you, Adira, and thank you everybody for attending. My name is Pouneh Dokouhaki, and I'm the renal pathologist. I'm pleased to say that I'm the new addition to the renal pathology group here, and I'm really, it's my honor to be part of this, the BC nephro/pathology community. Do you see my my presentation?
Yeah, but it's not quite in presentation mode. If you no, now I can see the second slide. If you go to display settings, yeah, perfect.
Better?
Perfect. Yeah, exactly. Thanks.
Just to start with the review of objectives, this is what we're going to do in this session. Complement system is an old innate immunity system, immune system. It carries the name of two giants in the field of immunology, Jules Bordet, which you may recognize from the Bordetella. The name was of the genus of the bacteria was coined after him. And he was the one who discovered the complement activity. And it was during the setup of an experiment to detect the patients who have been vaccinated and who has the antibacterial antibody against a certain bacteria. And he set up the experiment to add the serum of the vaccinated person or the sheep who has the anti-serum to the bacteria and saw that and observed that the bacteriolysis happens. And then once he heated the serum, that bacteriolysis activity disappeared. And then by adding a fresh serum from unvaccinated or sensitized animal, then this activity, that bacteriolysis activity came back to the serum. And because of that, they knew that there was a heat labile and heat stable component to the sensitization to the immunity against that bacteria. And he originally named that factor, heat labile factor, lectin. Paul Ehrlich is the second immunologist who's well known in the field of tumor immunology and the tolerance and all this stuff. And he was older than Bordet, but he came after him, and he used this methodology to actually follow up the patients with vaccination and form the complement assay that has been in use for hundreds of years. hundred of years after that in the hemolytic activity, and we will mention that later on. And he coined the name complement, actually. Complement system is an ancient innate immune mechanism. Its origin goes way back before the human or mammalian origin in the evolution. It is a complex cascade of molecular interactions, and all of them have enzymatic activity. They are produced as a proenzymes with no enzymatic activity, and then they get activated through a cascade of activation process. And then at the end, they produce bioactive fragments. What is really important to keep in mind when we talk about complements is all about balance. It's overactivation of this cascade can result in tissue damage, autoimmunity, and chronic inflammation, and underactivation or inhibition of this complement system, which we sometimes have to do it in a certain pathophysiological situation, will end up in reduced in immunity against microorganisms and infections, particularly with the capsulated bacteria, and also it can result in autoimmunity. So it's always good to keep in mind when talking about complements that the balance needs to be maintained. First and foremost, activity and function of the complement system is the immunity against bacteria or microorganisms. If you just imagine this yellowish green shapes at the top are flagellated bacteria. You see that the complement system has developed many ways of dealing with this bacteria before even the antibodies are generated against this bacteria. So this system is very fast and furious. So it acts really fast against the bacteria, and it's developed to do that job. And at the top of the screen, you will see the initiation part of the complement cascade. And for the ease of discussion and ease of study of this systems, very complex system, we divide them into initiation phase divided into 3 different pathways. And what are they different is basically the molecules that start the activation of the cascade are different in these 3 pathways. The first one is, which is more ancient one, is alternative pathway, and it's the initiation and trigger point is by activation of the C3 in the serum, soluble C3 in the serum, and recognizing the microorganism associated molecular patterns or damage associated molecular patterns on different cell types. So these are the molecular patterns that C3 recognizes even without the help of the antibody. And it just activates the C3 on its own. And also the constant activation of C3, we call it takeover, happens all the time in the serum, just spontaneously as well. This is alternative pathway, and then it produces the activated form, which is C3b. Lectin pathway, on the other hand, it just starts with the lectins that bind to the carbohydrates of the cell wall of the bacteria. We know that the bacteria are usually full of carbohydrates, and these carbohydrates moieties are not most of them are distinct from the mammalian carbohydrate moieties or patterns, motifs. So we have lectins, which is the name for the proteins that bind to the carbohydrates. So any protein that binds to the carbohydrates, we call them lectins. And then we have lectins in the body that's their job to just track any foreign carbohydrates and bind to them. And these are one of the most well-known of these lectins is mannose binding lectin, which binds to the mannose moiety on the surface of the bacteria and gets activated. So this is called lectin pathway. And then these 2 pathways are meant to act against bacteria very fast. even before the immune system started to recognize the bacteria. But then we have another pathway of initiation or trigger pathway that it starts with antibodies binding to the bacteria. So if the bacteria escapes these two innate pathways, then we have they're recognized by the immune system and the antibodies are being made against it. And then a complement gets also activated by activation, by binding of these antibodies to the surface of bacteria. And this activation and trigger goes through C1q activation, and it starts the classical pathway. Then all of these pathways, doesn't matter at the end, what is the trigger point, they come to a C3 convertase molecule. From the, this is like reverse, the picture is not right. From the classical and lectin pathway, we have C4b to C4b and 2b molecule that is a C3 convertase activity, and then from alternative pathway we have C3b and then two molecules of C3, and with the B factor B is just the consolidation molecule for this. Then we have the C3 convertase, and this convertase start what we call the amplification loop, where C3 convertase start converting and activating more C3 molecules into activated molecules, and then they bind and form the complex. And with addition of more C3bs to this C3 convertase, we will now have C5 convertase, and then C5 is generated. And then C5 will help to organize this membrane attack complex, or MAC, at the end, and it just starts the bacterial lysis that we can see with the different tests. Also, during this process, lots of bioactive molecules like C3a and C5a are produced. C3b itself has lots of effector functions. So in the bottom, you will see effector functions of this activation, whether through any of those arms of the complement pathway activation happens, we will see the same effector functions that going on. Opsonization, which means that the bacteria are tagged to be phagocytosed by the immune system. We will have the pro-inflammatory cytokines that call the immune system to come to the site of the inflammation. And then also they add the endothelial and epithelial activation and damage will add. On the side, also platelets get activated by the complement system. There is lots of similarities and homology between coagulation system and the complement cascade. So also the coagulation on the side gets activated. So it's a very complex. They just a start of the complement system will set off a huge amount of effector functions, all of this in the immune system and outside of the immune system. The same thing can happen to an altered cell, like will be recognized as an outsider, as a bacteria, and all these mechanisms can happen through the tumor cells, and that's why the complement is a crucial part of the anti-tumor immunity. I just want to emphasize again the complexity and interaction of the complement and coagulation system. Both of these systems are comprised of enzymes with serine proteases activity. So it's no surprise that lots of coagulation system enzymes helps to activate the complement independent of those complement specific triggers, and also the complement enzymes that are generated can help stimulate and initiate the thrombotic pathways. So always these two go hand in hand, especially if you have in the middle some endothelial cell injury and added elements of the platelet activation and other activation in the coagulation system. So we have to keep in mind that, you know, inhibition of one would result in inhibition of the other one, or activation of one results in activation of the other one. Newer roles for the complements are being recognized every day, and I think the complosomes, if you haven't heard of them, they're going to be the thing of the future, and it has been shown in a really nice, elegant studies that complement factor and those bioactive elements are formed even inside intracellular, inside the cells. And these elements, bioactive elements, are important for immunity, immune cell homeostasis, and constant activation. Because if immune cells don't see their cognate antigen, they tend to die. So that's how they're designed. And what we don't want to lose in the immunity, we don't want to lose their effector function. So by way of complement activation, this is a way to for intracellular constant stimulation, and then helps the homeostasis and metabolism of the immune cells. Both C3A and C5A, especially on the mitochondria, it has been proven to have the stimulatory effects for the metabolism of the cells. And C3 on the lysosomes, C3A has a special receptor and helps keep the T cells especially activated and produce low levels of interferon gamma and granzymes and keep those innate activities. Complements are really important in taking over or just clearing the necrotic or apoptotic cells, the cells that are of no use to the body. And this is the complement system and in the presence or absence of the inhibitory factors that when the cell undergoes apoptosis, it just make the path for the necrotic or inflammatory response or non-inflammatory response or silent removal of these apoptotic cells. And the complement and presence of the complement inhibition factors on the cell surface will determine which pathway the cell will be removed and what is the consequence of this removal for the tissue around it. One of the functions of complement that is usually overlooked is how much they help in solubilizing the immune complexes. If you look at the left side, it shows the chart, the graph that when you have in a state of adding antibody being present all the time, and you start to increase the amount of antigen for that antibody in the solution, this antibody-antigen complexes go through different phases. If you have lots of antibody and not so many antigens, we call them antibody excess. And in this situation, the immune complexes are usually small, they're soluble, they don't cause much of the trouble for the tissues and in the blood. But as you add more antigens to the system, then you come to the zone of equivalence or equivalence zone, where the antigen and antibody are actually of the same concentration, of the optimal concentration, and they start forming this antigen-antibody immune complex lattices. And this is what we see. It's pathogenic to the tissues. They can deposit on the endothelial cells and do some injuries. They can deposit in the mesangial cells of the kidney, and it's really important to avoid this kind of lattice formation and then aggregates of antigen antibody for preventing the immunopathology. And then as you add more of this antigen, you come to the phase which is antigen excess. And again, those immune complexes tend to break apart and become smaller in the antigen excess zone. What does complement do in the system is that by binding of C3b, as we mentioned, that C3b is or activated C3 is constantly generated in the bloodstream, and these activated C3s can bind to the antibodies and keep them away from forming these lattices, this huge aggregates of antigen and antibody. And this is very useful and very critical function of the complement activation in the system to avoid forming the immune complexes. Again, one of the surprising and interesting functions of complement, just they have functions all over the body, is just a regulation of synaptic formation in the brain. And with this, again, they form this tagging system that tags the synapses for phagocytosis by the microglia in the brain to get rid of those synapses. So they have a tagging that says, oh, this neuron system is not useful anymore. Just get rid of it. And this tagging system works very efficiently. So the complement system now, it's one of the factors that are considered in neurodegenerative diseases. And we know that in retina, for instance, in macular degeneration, there is a role for the complement, and complement activation, if it's blocked, can block the progress of the disease. Complement system, just briefly, I mentioned that they have really a wide array of receptors on different types of the immune cells particularly, and through these receptors that these different bioactive fragments of complement, even after deactivation, even after they don't have any enzymatic activity, just the remnants of those complement molecules, they will have some additional function in regulation and activation of the immune system. And this is like huge field that still needs to be explored more. So they can, by just binding to these different receptors, they can basically stimulate and modulate the activity of many immune cells. One of the new areas that might be of interest to the kidney pathophysiology is that recently they have been found that, like in mouse B cells, by binding to the complement, because of the difference in the receptor aggregates or B cell receptor kind of complex that they form on the surface of B cells. For long, we thought that the complement activation will increase the activation of the B cells, and this is based on the mouse models. But in human, they found that because of this, the way the complexes of the BCR, B cell receptor, and the complement receptors are different from the mouse, then most of the time the binding of the complement activation fragments to the receptors on the surface of B cells will end up in inhibition of the B cell activation. So we need the complement activation to keep the B cells on check. So this is really important. So when we think about inhibiting the complement altogether by the medications or by different mechanisms, then we unleash the effect of the B cells activation. So we will have more B cell activation and more immunoglobulin production, theoretically, in the context of immune complex diseases. I just want to bring this to the attention just briefly. Again, this is the different diagram of the same pathway. We have classical pathway, activation of the complement through the antibody binding and C1q. We have lectin pathway, activation happens through the mannose binding lectin, MBL, and then and these two pathways go through the activation of C4 and C2. And then we have alternative pathway, which is like a very rapid response to the all the stimuli that we mentioned before. And then C3 activates with the help of or consolidation with the B factor B will end up with the amplification loop. I just want you to pay attention that after the complement gets activated, for its effect to be activated on the cell surface, they have this complement fragments, they have to bind on the cell membrane. So then on this is what is called, the process is called fixation of the complement. So the complement factors, when C4b and 2b are generated, they just start binding. They have a binding availability appear after this kind of the molecule opens up, and they bind to the surface membrane of the cells of interest. And it's really important to keep that in mind that the C3b and BB, they don't complex in the fluid phase. And again, just mentioning of the fluid phase and the membrane phase or the tissue phase. So that's where the terminology comes from. So activation happens in the fluid phase, in the serum, but then the products of this activation, they need to be fixed on the cell membrane to continue to have this amplification loop and then start to have the C5 convertase. You need to have this the complement components or C5 convertase on the surface of a cell membrane. And it's important because we just want to mention later on the test that we have available. So, and then the membrane activation or membrane attack complex should be on the surface of a membrane. And that's what. Another factor that we have to pay attention to is that on the surface, when you have the either classical C3 convertase or alternative pathway C3 convertase, which is BB and C3b, then they will form the C5 convertase by addition of more C3s. So it doesn't happen that usually it just those algorithms that we see in the books and textbooks just gives us the impression that only one molecule of C3b addition to that is enough for getting the activation. It's not true. Actually, the more C3bs are kind of added to this the C5 convertase activity, the activity of the C5 convertase would be more. So you need a lot of C3bs to get deposited in the same place as you have this the combination of C3b and BB and C4b and C2a. These complexes, they need to be complex to many C3bs before they can have the C5 convertase activity. And this is important because just the amount and the intensity of the C3b on the tissue matters for activation. If you have a little bit of deposition, it might not have the same activation consequences as if you have a lot of C3b. So it's a game of density, basically, of these molecules on the surface. And it just signals, again, going back to that balance, if you have a lot of cells, C3b activation, it just gives you the signal that the complement is still activated, the inflammation is ongoing, and so on. But if you have a little bit of C3b, it means that the complement activation didn't happen. So what do we have in terms of checking this complement system in the laboratory? So we want to measure this activation. We want to, if you want to control them, you want to have a good test to measure the before and after of the regulation. Unfortunately, we don't have lots of good tests. The test that we have basically goes back to this 100 years ago that they have those bacterial lysine. We check for the activation of the complement in two different phases. As I mentioned, you have a fluid phase and we have a tissue phase or fixed phase. And we test the complement into two different categories. If you want to use them as biomarkers, we should be very careful because in addition to biological plausibility and then the response to the regulation that we make by the treatment, they have to be easy to measure and they have to be a standardized methodologies. So sorry, I just want to mention, so by the tissue phase, basically we see them on the tissue. C4D is a really good biomarker of complement activation. I will mention later on. And then in the fluid phase, we have different tests that I will mention. So for the fluid phase, we can measure the complement levels at the just protein levels, total protein levels, regardless of activation or not activation. And we do that, we measure that by nephelometry, usually on the big chemistry units. They have very limited sensitivity and specificity, and the reason is that they have a wide range for reference range, because you can imagine the complement system, just the amount of protein, these are acute phase reactants. There are lots of variation between individuals, and even in one individuals, you will have variation depending on the level of inflammation that's going on. So I think the most important one when you're checking the total protein is the trajectory, and the repeated measurements are important than the just one-time measurements and it's low or high. So the sensitivity is very limited. Then it comes, because of that, we need some functional assessment of complement. Traditionally, CH100 or CH50, and then alternative H50, and then we have lectin, hemolytic activity 50. So these have been traditionally used. They're very hard tests to develop and standardize. Now most of them are replaced by complement activation assay. I will explain in the next slide. But also we can test for the individual proteins, C1q, C3, C5 in the pathway. And again, they have the problems of the total protein measurements because we don't measure the activity. One of the important factors are the C3 nephritic or C5 nephritic factors, and these are autoantibodies, and they can be measured like any other autoantibodies. They're good tests available for them. When we talk about the functional assessment of complement, this is how the testing is usually done. We have sensitized erythrocytes or red cells, and then you add the serum and see how much based on the complement activity, you see a more or less the hemolysis. This was very hard to control, hard to standardize this test. Now this is alternative way of testing, very similar pathway by liposome assay. And then liposomes are in vitro made particles that have the same ability as the red cells, and they can, by binding to the immunoglobulins and then complement activation, they release G6PD, usually the intracellular enzymes, and then the enzymes activity, we can detect it by automated systems. These are hard to control for the positive and negative control, but nevertheless, they're automated systems. How do we measure the activation fragments? This is another way that if you say like just replace the CH50 or AH50 activities by the hemolytic assays, and we have different pathways like classic pathway, lectin, or alternative pathway, and the coating on the bottom of the wells are different depending on which pathway we want to test, and you add the serum, and based on the fragments that are generated after the activation, we just measure them and have an idea, basically. So they're not really, doesn't emulate the complexity of the complement activation in vitro, because complement activation happens at the same time. Complement, the alternative and classic, they have to happen together to be more of the same activity. So they're just poor standardization. There's just very much conflicting data that comes if you measure this, especially the soluble C5b, C29. This is soluble MAC marker. They usually use it as a marker for complement activation. The problem is that, as I mentioned, most of this MAC activity is on the surface of the cell membranes, and they're fixed already. So you don't see them much in the circulation, and they're not a good indication for how much activity is going on the membrane. And then another factor, some people do this complement activation in the urine. Again, these are not standardized. They're just, we don't know whether the fragments that we detect are from the systemic inflammation or are they from the locally produced in the kidney. And also, there are just some activation happens in the tubules as well. So what do we have available for the tissue phase? We have really good biomarker as C4D measurements, and it has worked very well in the assessment of complement activation or antibody-mediated rejection in the context of transplant. And it works very well because there has to be an inflammation going on for a long time and factor I be activated. For the C4, when it gets activated, it binds to the membrane. That's what we said, complement activation binds to the membrane. Then you need a continuation of the inflammation and the factor I involvement for the C4b to be broken down to the inactivated C4b and then generating C4d. So you know that up until the generation of C4d, complement activation was constant and was present there. And then before presence of C4D, like this epitope or this antigen is not exposed until the very last minute. So you're sure of the complement activation happens when you have the detection of C4D by immunofluorescence or immunohistochemistry on the tissue. We have problem with C3, so we cannot repeat the same idea with the C3 deposition. There are many reasons. One reason that we see generally is that even in the context of C3 glomerulopathies or C3GN, that dense deposit disease, confirmed dense deposit disease by EM, we still see, so these are the A to D, are different cases of dense deposit disease. And they can have from 0 C3 deposition to the very high, like 4+ deposition of C3 in the glomeruli. So it just tells me they're just a problem with the sensitivity of the C3 using of C3. And more problematic issue is that we have lots of false positive of the C3 deposition in the tissue, which is not as a direct activation of the complement. One example of this, we see C3 deposition all the time on the IF samples that we examine in the lab, and sometimes it's because of the hyalinosis that is available in the arterioles. Activation of the endothelial cells might end up with the deposition of C3, which is, we don't know what is the significance of that. And these are lots of non-specific pattern of injury that we see C3 deposits there. And the reason for it is that with the way we detect the antibodies that we have in the lab available, it's for C3c fragment. And C3c fragment is common in the inactivated C3b. You go up to the channel of the complement activation. C3c fragment is also available in C3b and also in C3. So with this antibody, this is in contrast to the C4d antibody in the tissue. When we use the C3c, we think it's a C3c antibody and we're detecting the complement activation fragment, but actually we are detecting all C3 molecules altogether. So the better way would be if we had any C3d or C3dg antibodies, that would be a better indication of complement activation. So with this, we really cannot claim that if we detect C3 in the tissue, it's as a result of the activation. So we have tricks to use. If you remember, I mentioned that the intensity of the C3 on the membrane deposition is really a critical factor for taking over the complement activation to the C5 convertase. and activating further. So if we see a lot of intensity score, we use intensity score to imply that it is activation. Also, we use internal and external controls. Pattern of positivity is important. Correlation with other studies are important too, but these are very like manipulations that we do, and because of that, this is not a good measurement as a biomarker. I just want to mention this, just problems in the testing and measurements of the complement. Sorry, it took me a long time to get over this, but is there any questions so far? They can go to the kidney and the complement.
I think a lot of things got resolved in the chat, so if you want to go on, that'd be great.
I'll just take a, is there any question? No?
Doesn't look like it.
Okay. Sorry, I might have confused everybody. So in the past few years, I would just say in the past year alone, many reviews and papers have been published in the field of kidney and the complement. And the reason, I think the common denominators of them is that they want to move from rare disorders to more common disorders with the complement treatments. Some of this might be a pharmaceutical push because now they have good drugs, but they only have the target population is a very rare diseases that are out there like C3GN or atypical HUS or paroxysmal nocturnal hematuria. So they want to have more customers. But I think this is a good sign for the academic community to take advantage of this enthusiasm to further our understanding of the complement. So many complement can have direct or indirect effect on the kidney and its diseases. In healthy state, most of the endothelial cells, particularly because they come into the fluid, in contact with the fluid phase of the complement, they have many inhibitory receptors like factor H or CD46, and these are what they call "don't eat me" signals that they give to the complement system just not to get activated inside. What happens is that in disease states such as C3GN or atypical HUS, these are very rare diseases, but they're prototypical complement-mediated disease activity. Those inhibitory signals are get either inactivated by mutations or the complement gets more aggressive. So they overcome those inhibitory factors that are available at the surface of the endothelial cells. I just want to go over, move quickly because of interest and in the interest of time. ANCA-associated vasculitis is one of the diseases that it's autoantibody mediated. We generally call it pulse immune, but it has been seen in 60 to 70% of ANCA biopsies that we have some deposition of complement. And there have been some very elegant preclinical studies in mouse that shows that C5 deficiency or blockage of the C5 receptor were protective against this ANCA activity. And they think that even though the ANCA pathogenesis is through the these autoantibodies against MPO and PR3 on the surface of neutrophils, but the additional activation of the complements potentiates this this pathway and this pathogenesis and tissue damage. And then because of those preclinical studies, there was phase 3 clinical studies published in New England Journal of Medicine 2021 that they showed that at least the when they treated with avacopan, which is a C5 receptor blockade, they showed that there was the results was non-inferior to the combination of rituximab and or cyclophosphamide plus prednisone. So they replaced prednisone with this avacopan, and they showed non-inferiority, plus the less exposure to the prednisone, which is, I think, this additional benefit for the patients. And because of that, FDA approved the use of avacopan in the context of ankle vasculitis in 2022. For complement-mediated glomerulonephritis and tissue injury, it's a more complex situation because you're initiating process is through autoimmunity, and you can block this autoimmunity with the good immunosuppression that we have, and only complement activation comes as a secondary wave and a secondary factor. But again, because we want to, pharmaceutical companies want to move to more common immunocomplex generated or diseases, IgA nephropathy was like the ultimate goal. So it was very nice. You know, the pathogenesis of IgA nephropathy is a 4-hit. Usually they call it 4-hit. Pathogenesis is that now we have like very galactose deficient IgAs in the serum needs to be increased, and we will have the antibodies against this galactose deficient IgA, and they form immune complexes. In this situation, I think the complement system will help breaking down this chains or these aggregates of the immune complexes. But these immune complexes can get deposited in the mesangial area and subendothelial and do tissue damage in the kidney. So where does the complement pathway fit into this pathogenesis? Is that you think that C1q needs to get activated with the immune complex deposition, but it's not the case in the case of the IgA nephropathy. Usually lectin gets activated by this polymeric IgA and IgG deposition. And the reason they thought is that the dominant immunoglobulin is IgG2 in these complexes, and IgG2 doesn't fix C1q very nicely. But because of this tissue injury and because the accumulation of the extracellular matrix, MBL binds to these immune complexes that gets activated. And there has been a chain of publications, I think it started in 2010, going forward, especially coming from the Asian countries where they have more IgA nephropathy, that they see this kind of deposition of C4d in the mesangial areas of the IgA nephropathy individuals, and they find it directly relevant to the cases of IgA nephropathy with a high MESA score, and especially in the crescentic forms. And then interestingly, so this is what do we think that MBL or mannose binding protein and complement causes the problem and causes a tissue damage. But interestingly, genetic absence of MBL actually predispose the patients for more severe course of IgA nephropathy. So it seems to have a U-shaped effect where its absence causes the problem, and because of all the good effects of the complement that we mentioned on the immune complex deposition in the tissue. So if you don't have the complement activation, you will end up with having more deposits and more activity and more tissue damage. If you have less, if you have less of this complement activity, you also have tissue damage and tissue injury. So this shows the complexity of the interaction of complement with the kidney diseases specifically. And other areas of intense research, I think, again, because they just want to explore whether there is any benefit from using this medications, complement inhibiting medications in the more common diseases, diabetic kidney disease and FSGS. the direct involvement of complement has not been really shown in these two diseases, but there are hints that complement activation, particularly through the mannose binding pathway, would increase or would add or potentiates the already pathogenic processes that are in place. Now, I briefly mentioned at the end, like the therapeutics or the complement system or reducing the activation of the complement that are available. There are a few that have been approved, and we know eculizumab, ravulizumab, and avacopan. I mentioned that, and there are also some medications like for the inhibition of the triggering point or the upper pathway. So this is the list of the approved ones. There are not many. What relates to the kidney, there are phase 2, 3 of the testing those inhibitory factors for the C1q or MASP proteins in the ischemic reperfusion and kidney transplantation. C3 glomerulopathy, we all know those medications, and also the avacopan, that they have been approved for the ANCA associated, but there are studies in phase 1 or 2 for the lupus, C3GN, and the IgA nephropathy as well. For every drug that is approved, at least 4 or 5 drugs that are in the pipeline, and I think this is a good sign, but we have to be very careful when we don't have a good lab method for monitoring and for assessing the complement activation in this situation. We should be very careful with going forward. And this is by this I end this presentation. I thank you for your attention and your patience, and I welcome any questions that you have.
That's great. Thank you so much. Like, that's a wonderful refresher course for some of us and new knowledge for others, and really exciting time to think about as we appreciate more the role of complement. I think we always thought it was there, but I think we always knew it was important in specific diseases, but I think you've highlighted how it's probably much more ubiquitous. One of the questions is talking about triggers of immune diseases. Are there available tests for predictions to start preventative treatment? Kind of provocative.
Yeah, like in terms of IgA, or because most of the time, especially in the kidney, complement activation is a secondary pathway or just additional method of or additional factors for pathogenesis. The only 2 examples that we have that C3s are just complement activation can cause the damage itself is the C3GN and AHUS. The rest of the entities, complement is the additional factor. So I don't know in prevention they mean prevention of C3GN and AHUS or other.
I think people are looking for like early predictors prior to the actual evidence that the immune system has been triggered. I think that's beyond, I think, current technology. And then I guess just we don't have a lot of time for another question unless people want to raise their hands. So yeah, Sean, you have your hand up. Why don't you ask a question?
Thank you, Pouneh. That was a great talk. I'm intrigued by in the first part of your talk, the link between complement and inhibition of B cells. And so, as you mentioned, there's been a lot of push to study complement inhibitors in a variety of immune complex mediated diseases. In IgA nephropathy, we've been part of trials in looking at like the Ionis drug that inhibits complement factor B, or the cemdisiran drug that inhibits C5. In all cases, we've noticed a pretty rapid recurrence of disease when you stop the therapy. Is there a possibility we're shooting ourselves in the foot if we inhibit only complement? Are we in a situation where the B cell activation may increase? We actually may have increased galactose deficient IgA1 levels as a byproduct of inhibiting complement. And do we need to think about combination therapy in those circumstances? And has anyone measured GDAG1 levels, in complement inhibition?
Exactly. This is exactly what I'm afraid of, because if you know the basic pathophysiology and physiology of the complement activation, you're really scared of just blocking the complement altogether, because it has such a regulatory effect in all aspects of immune system. I didn't show also the role of the complement in T cell biology. It's just huge literature available right now. And B cell activation, you're absolutely right. And that's why we should see this complement. So we tend to see the complement in a very linear way. It is pathogenic. Activation happens this way. And if we detect activation, we should block it, and it reduces the pathogenesis. But I think it's a very simplistic view, and it's because we don't have much insight and information from the basic biology yet available. But yes, the B cell inhibition through the complement receptors is something very new and something we didn't know before, because the mouse biology is different from human biology. And this is maybe in the past 4 or 5 years that came to the light, that, okay, the complement system actually inhibits the B cells, because it makes sense, because you don't want an inflammation to go on forever. You want the B cells to, when the complements take over, it just reassures the B cells that, oh, there's not much to react against now, just quiet until I finish my work, and then you can come and see whether there's any pathogens or any other stimulus available. It just makes sense to me in the context of immune biology.
Great.
So we should see this as a complex system. So yeah, that's exactly right. We might do more harm than what we think.
Great. Thank you. I guess we're at the bottom of the hour, so if people have questions, I think it's fair to say that they can send them to you directly or call you. But welcome in advance to BC, and looking forward to ongoing interactions with you and the rest of the pathologists. We're learning a lot and understanding so much more than we did even 5 years ago. So thank you for that wonderful presentation.
Thank you, everybody. Have a good day.